My name is Scott Manalis. I'm a professor at MIT. I'm in the Department of Biological Engineering, and my lab is in the Koch Institute for Integrative Cancer Research. Broadly, my lab is interested in developing new instrumentation for cancer research, and outside as well, other types of areas of life science, but mostly focused on cancer. And we do that primarily by developing new approaches for measuring biophysical properties of single cells. And we have ways to do this with very high precision that I will show you in my presentation. So the quantity that we have been measuring from single cells that we do really well is mass. We can actually weigh single cells with just this incredible precision. Then I want to give you some context for this. So the ability to weigh things has been of interest to physicists and electrical engineers for quite some time. And there's really two things that have made this attractive as a fundamental measurement. One is that so first, let me explain what you're looking at here in this picture. What you're seeing is it almost looks like these little overpasses connecting these two side regions. And those are suspended bridges. You could almost think of them like a guitar string, a very short string. And those things, if you shake them at their resonant frequency, will vibrate. And if you put mass on those things, the frequency will change in proportion to how much mass you put on it. Now, people in the area of electrical engineering and physics have been able to make these things incredibly small. This is really the area of nanotechnology. And the smaller you make these things, the less they weigh, and the smaller amounts of mass that you could actually discern. So nanotechnology has allowed people to make these very, very small resonators. Number two is that electrical engineers have figured out how to measure frequency with just this incredible precision, down to about very close to 1 part per billion. So you take this small object that you've made, this vibrating guitar string, you divide this by a number close to 1 billion, that tells you how small of a mass that you can measure. And so this has been kind of a race over the years to push the limits down further and further. And actually, the record now is at the level of yoctograms. So what we're talking about here is individual gold atoms that bind to these guitar-like strings, changes the frequency enough that can be measured and detected. But what we're interested in is in the area of life science. And there you have to do this underwater. And all of this work has been done in vacuum. And it's needed to be done in vacuum so that these resonators can vibrate with just a really high degree of fidelity, like the high quality wine crystal. But if you actually take this underwater, this situation completely changes. There's a viscous damping that makes the frequency resolution just go down by orders of magnitude. And so what you're left with when it comes to bringing this underwater, the limits have really been around a nanogram. So far, far, far away from this level of individual gold atoms. And to kind of put that in perspective, the weight of a mammalian cell is about that. It's barely enough to weigh a large size mammalian cell, much less be able to look at small changes in it. So what my lab did a number of years is we took this idea of a resonator. In this case, I'm showing one that's in the shape of a cantilever, where mass binds and changes the frequency. And we made it hollow. We actually put fluid inside of it. And so we got rid of the outside viscous drag from this. And then we can flow cells inside of the cantilever and measure frequencies as we do that. And this turned out to improve the detection limit by 6 orders of magnitude. It took us from nanograms to femtograms. So now, all of a sudden, not only could we weigh a cell, but we could look at very, very minute changes in how much a cell weighs. And this has had really big implications that I will show you in terms of being able to read out fundamental properties of cells in their environment and how they respond to drugs. But first, I'll just show you that these devices we've have been working on for many years to fabricate them. They're actually within a vacuum package that's on the chip. So these things are made on 8-inch wafers up in the top left corner. And each chip now, if you go to the middle picture, is the size of a postage stamp. And if you zoom into those, you can see the little diving board there. You can't see the channel inside because it's sandwiched in silicon, but that's vibrating in a vacuum cavity. And you can see little electrode contacts above that we use to make contact to it to be able to measure its frequency. So here is a little bit more closely showing you how this works. So this thing is vibrating. We flow cells one by one through them, very similar to the way a flow cytometer works. That little dip downward in the frequency is how much the cell weighs in the solution that it's in. So we call that the buoyant mass of the cell. If the cell had the same density as the solution, there would not be a signal there. So it's how much the cell weighs in the solution that it's in. And so to give you an idea of the precision, what we're doing in this experiment is we're taking the same cell and we're flowing it back and forth by controlling the pressures through this cantilever. And each time we measure the height of the peak and we plot that. That's a point on this plot here. And so this gives you an idea of the scale. So 1 picogram of mass is like a 75 nanometer increase of the cell. This is a mouse lymphoblast. And if we look at our noise level, so our noise level puts us at less than 50 femtograms, or 0.1% of the cell mass is what we can discern. And so in terms of position, that corresponds to the cell changing its diameter by about 5 nanometers. So that's what we could actually resolve with this. So this is more precise than even really advanced forms of microscopy can do by looking at these very small changes of frequency of this device that we can measure just with this high degree of precision. Now, over the years, we've been able to show that this device can measure more than just mass. For example, in this recent work that I'll tell you a little bit about pretty soon, we can excite acoustic waves within this device that actually probe the mechanics of the cell. It changes the fluids in such a way that it perturbs the cell by just tens of nanometers and squishes them in and out. And we can measure the stiffness of the cell from doing this in a very non-invasive way and very high throughput way. So that's one example. Another one is that we could actually measure the density of the cell, and we can measure its molecular composition by weighing the cell in different fluids, and from there directly calculating essentially the dry density of the cell, the total density of the cell, and the dry mass. So we could get a lot more very fundamental parameters about the cell from doing this. We can also take the same cell and weigh it multiple times, kind of like I showed you in the slide before, but we have devices where they can flow through and get weighed multiple times in one pass as they flow through, and we call the output from this mass accumulation rate. And then with all of these methods that you see here, we have the ability to take cells off the chip, put them in wells, and link each well to what we measured before, whether it be the growth rates here, the acoustic scattering, density, whatever we have. And then this is compatible with downstream single-cell RNA-seq. So we can actually link these biophysical properties to the transcriptome of the cell or any other single-cell downstream assay that you would like to perform. Okay, so let me tell you now some of the applications that this has got us into. So first, I want to go to bacteria. And so we're taking this device now that we made where we have strung together several of these resonators in one long fluidic channel. So a cell comes in and gets weighed several times as it travels through this. This here is the trajectory of one cell that you're seeing in this green here. And if we zoom into that, you can see there are several points from each weighing of one of those devices. Now that's one E. coli traveling through. We can take the slope of that, and that is what we call the mass accumulation rate, or the MAR. Now we can plot that, and we can flow many of them through at once. And you can see the individual mass accumulation rates of these E. coli. Now watch what happens when we put a drug into this. So below now, at time equals around 80 minutes, we are putting an antibiotic into here called kanamycin. And immediately following there, you can see the slopes start to reduce and become flat. And the mass ultimately goes down too. If you look towards the right, around 140 minutes, you can see the mass is clustered at a lower level. So there's a few important implications from this. One is that we're looking at the response of this antibiotic, and we're seeing it on a time scale of tens of minutes. And so by having this high precision of mass, this really translates to the ability to have a very rapid readout for drug response, much quicker than it would take if we wanted to use a conventional way, which looks at proliferation and essentially use light scattering to shine that through a collection of bacteria. And as they stop dividing as frequently, you can read that out in the way the intensity of the light scattering through. In this case, since we're measuring individual cells with this precision, we can see this much earlier on. And it turns out that has huge implications for being able to make what's called a rapid antibiotic susceptibility test. Right now, these tests take several days to do, and in the ICU, that has huge ramifications. And what I'll show you is that these can actually reduce that time down by a factor of 3. And then second, these are single cells, so we actually see the heterogeneity. You can actually see differences across different cells. At one given time point, some cells change their slope a lot and some a little bit. So we get this look into the heterogeneity of growth rates across cells. And then finally, all this is done without consuming many cells. So we can take small number of cells to make these measurements. Again, for practical applications like antibiotic susceptibility testing, one of the reasons why this takes so long in the clinic is that you have to grow up enough bacteria that you can scatter light through it and see something. But in this case, we can make measurements in very small numbers of bacteria. And so I won't go too much into the details of this, but I will say that I co-founded a company. So as full disclosure, I'm a co-founder of Affinity Biosensors and have equity interest in the company. But I will show you here that they have made this instrument, kind of a desktop instrument to do these rapid antibiotic susceptibility testing. And inside there, you can see if you load in a 96-well plate that has the drugs preloaded, and then it's read out kind of well by well. And what you're seeing here in that plot is how in about, in this case, several hours, you can start to detect the growth of bugs in some wells, but not in others, to actually read this out. And so this company has submitted to the FDA what's called a 510(k) clearance last summer. And with that being approved, this could actually then be used in hospitals to return results directly to patients. So we're very excited about this development. Okay, so I'm going to change now to a different application that we've been pursuing. So we're going to move from infectious diseases to cancer. And unlike infectious disease, like bacteria-based ones, where you can do very clear tests to know which drug is best for the patient by directly testing the drugs on the bacteria from the patient, for cancer, it's actually really different. And in fact, for most cancers and most patients, the drug ultimately ends up being chosen empirically. Now, there are some cases where there are biomarkers that can guide this. And in those cases, it's really done based on genomics, based on sequencing, genomic profiling of the patient. And that has been the most successful approach in the clinic to be able to match patients to a drug. But the problem with it is that for many patients, when they sequence, there aren't mutations that are actionable. In other words, mutations that tell the physician that this is a drug to use. In fact, on the bottom right here, these are results from a recent study. And out of 5,540 patients that come in, only about 12% were actually able to get prescribed drug based on their sequencing information. So this is a very powerful technique. It's the best thing now in the clinic for guiding therapy, but for most patients, it's not accessible. And so this is something that we're interested in changing to have assays that can be complementary to the sequencing to widen out the number of patients that could have therapies chosen for them in an effective way. And so one approach for this is called functional precision medicine. The idea is you actually take tumor cells from the patient, you take them ex vivo, and you challenge them with different drugs. And then you look to see how those cells respond to the drugs. You measure the ex vivo drug response, and based on that, then you make a selection, in this case, the green drug, for that specific patient. Now, this is an idea that's several decades old, and it really hasn't worked out so well because it turns out that cancer cells are more complicated than bacteria, and the drugs don't always have the same efficacy. And so what you're seeing on this slide is what's been done for bacteria, but as it is, now it has not worked so well for cancer cells. But over the past 5 to 10 years or so, there has been a number of labs that have re-entered this field, including my own, kind of a resurgence of this to attack this in a new way that is showing promise to go beyond what had been done before, where essentially the cancer cells were just plated on a dish and kind of treated in a very simplistic way. So let me show you, there are two strategies now that are being pursued to address this problem. Okay, so one is you take a biopsy from a patient, you disassociate it, and then you actually seed it as organoids, and you grow it up into more complex structures than they're just plating these cells on a plate. And the ability to make better and better organoids has really progressed a lot over these years. The idea is that can more faithfully replicate the tumor microenvironment. But still, with this approach, a major challenge is that you need to be able to engineer the tumor microenvironment to maintain the same cell state that it was in the tumor. But this is an area that many research groups have made significant progress on and is starting to become a possibility now. And then you have the ability to do extensive drug testing on all these cells that you then have essentially grown up to not only think about selecting drugs, but to be able to find mechanisms for resistance as well. Now, one downside of this approach is it takes a while. It could take several weeks and sometimes a month or more to make this material. And that's not always clinically actionable to have to wait that amount of time. So a separate strategy is what's called acute drug sensitivity testing. And so you take them much earlier on, before you even really proliferate these cells. So they've had minimal drift in the cell state. And you challenge them there. And so there, the advantage is you actually can return results rapidly to the patient within days, which, based on many discussions we've had with oncologists, really makes a big difference. But there, the challenge is you need to have early indicators of drug sensitivity. If you just simply try to measure proliferation, like has been done for bacteria, that doesn't always work because the cells are kind of on a race to death, even if they haven't seen the drug in this early state. Many of them are dying. And so you need to have readouts of the cell that can tell you what the drug response is. So that's what I wanted to talk about next. So I'll show you one example of how we've been using mass to do this. And so this is a collaboration that I've had for quite some time with Keith Ligon and his lab at Dana-Farber Cancer Institute. And what we did in this study is that Keith's lab is able to generate organoids from glioblastoma patients. And we took 69 of these patient-derived models that you see here. And we challenged them with a drug called TMZ. This is a standard of care that almost, not all, but many patients will get that have glioblastoma, brain cancer. And we then took a control set of cells and gave them a control DMSO drug. We then took those two different samples. We disassociated the cells into single cells. We weighed them with this cantilever structure that I showed you, and we measured the distribution of those cells. Now we're kind of at the bottom left there. And what we found in the study is that patients who were responsive to TMZ, their mass distribution of the drug-treated cells were different than the control. Whereas patients that did not have a response, whether they're drug treated or not, their mass distribution were identical. You can see that in the bottom histogram there. And so what we did, what was very unique in the study, is that we had patient outcomes. We knew overall survival for each one of those patients. So we asked the question, does this shift in mass distribution that we see, does that predict overall survivability? And what we found is it did. And so that was very exciting for us. And this happens to be one cancer where there is a biomarker based on sequencing that's called MGMT promoter methylation. And we did as well as that biomarker. But the message from this is that if it works here, perhaps it could work for other cancers where there does not exist a sequencing-based marker. And that's a very active area of our lab right now. Okay, so I want to go on to talk about a different area here, and this is immune cells. So as some of you might be familiar with, a lot of the new exciting drugs developed recently are aimed at really improving the ability of the immune cells to target the tumor. And immune cells are something that we've been able to also make very precise measurements of. So what I'm showing you here, these are T cells that we have taken from human, and we've activated them ex vivo. And what we're plotting here is the mass of each cell on the x-axis, and the y-axis is this mass accumulation rate, is the MAR. And you can see at T equals 0, these are mostly resting T cells that are just kind of sitting there. They're very small. They're mostly just the nucleus. They haven't really had a chance to grow yet. But over time, once they have had this activation signal, you can see they start to get bigger and they start to slowly accumulate mass. That's at 12 hours. Now, at 24 hours, you can see they've gotten bigger and are accelerating more in their growth. And by 36 hours, you see kind of there's a spectrum of mass sizes here. And at this point, they're starting to blast. They're growing and dividing. So we've been able to have a very precise way to follow the activation of T cells by their mass. And so one of the areas now that we're looking into is to what extent could this information tell us about the performance of the T cell activation and any dysfunction in the T cell itself. These are very preliminary results. that I'll show you. But what we're looking at here these are from patients. These are severe T cell dysfunction in CLL, chronic lymphocytic leukemia. And what we are able to see on the left, actually, is if we compare these CLL samples in red, they actually have a different mass distribution than the healthy counterpart. And when we activate them, their ability to accumulate mass, which is shown on the right, is also different. They can't accumulate mass quite as well. So we're very interested to use this generally to look at dysfunction in T cells and to look at how immunocheckpoint blockade therapies will alter the ability of these T cell size distributions or mass distributions that we can detect, and really ask the question, can we be able to predict how a patient is going to respond to one of these immunotherapies by looking at these T cell mass-based measurements. So finally, I'll just show you that I have co-founded a company called Travera. Again, in full disclosure, I'm an equity owner in this company. And what you're looking at here is a room full of 24 of these instruments that I've been showing you results from. And these are now in a CLIA lab. So the lab has been approved so that it can actually now return results to an oncologist, and the results can be shared with the patient. And so the way this lab works is typically around 5,000 cells per drug can be tested, 30 minutes per drug to test. And what's really important here is a 2-day turnaround. And so now the main goal is to figure out to what extent can this predict patient outcome. And so there are clinical studies now being run on these systems to be able to answer that question. So I want to tell you more. I want to tell you now how we can get additional information from these cells besides just their mass. And so what I'm showing you here is how we could actually measure the density of cells. So here what I'm plotting is the buoyant mass as a function of fluid density. And look what happens if we weigh the cell in a different density. So in green here, the fluid is percol. So the density is 1.1. The cell actually floats here. And you take that measurement, you connect those lines. The intercept is the density of the cell. The intercept of the y-axis is the total mass. Now, if we weigh the cell also in heavy water, and so this is deuterated water that exchanges very rapidly throughout the cell, we're essentially weighing the dry material, and it sinks like before, but not quite as much. This tells us the dry density, which is purely based on the material composition of the cell, and the dry mass. So by weighing these different fluids, we can gain quite a bit of more information about the cell. So I'll show you just a couple of things that are interesting. First, I should show you how do we exchange these fluids. So here's how we do it. I'm going to play a movie to show you. So the cell goes through its weight in one fluid, and then the higher density fluid, say, is in yellow, and now the cell returns in this higher density fluid. And so that way, by flowing the cell once in one fluid and then returning it in the other, we can get 2 measurements of that cell in 2 different fluids. So I'll show you what this looks like. So we're plotting here the mass versus volume of cells that we measured this way. So this is from a cell line. The bottom left there are cells that just divided, and the top right are ones that are about to divide. But look what happens when we plot now the density of the cell as a function of its mass. You can see that the density is very tightly conserved, actually. So if you actually look at those numbers, the coefficient of variance of density is almost 2 orders of magnitude smaller than mass. And so cells, it turns out, are very tightly regulating their density. And that has made it a really interesting biomarker to look at, because that regulation changes when you perturb the cell. And I'll show you an example. So these are cells that we've treated with staurosporin. It's a very strong drug. It's a broad spectrum kinase inhibitor, but this is to illustrate a point. And when we treat these cells for just actually a few hours of this drug, what you see here is that there's drastic change in density. Turns out this is just because of water content. The cells are getting rid of a lot of water, and it makes them more dense. And you can see the density shoots up. Their buoyant mass hasn't changed at all, actually, but the water content has. Here's another example. Here, okay, so same thing here, we're plotting it versus volume. So you can see there is a shift in volume because the cells have gotten rid of some water. Okay, so let me show you one other drug here. This is rapamycin that targets mTOR. It's an mTOR inhibitor that's critical to growth and nutrient sensing pathways. And here the effects are more subtle. So you can see in red are the cells that have been treated with this mTOR inhibitor, rapamycin. And so their density has changed. And if we look at this, so what we're seeing here is this is actually the equation for buoyant mass. So there's some change in density, but there's also change in the material properties, the composition of the cells that have contributed to this. And so this is another kind of way to see that with density, we can see very subtle changes. In this case, that would be difficult to see by just mass or volume alone. Okay, so the last part of the talk, I wanted to move on to what will be the third and last application here. And I'm calling this targeting minimal residual disease in cancer. So MRD, this is the idea that you treat a patient, and particularly with blood-based cancers, a lot of these drugs are so effective that 99.999% of the tumor cells are eliminated. But there's a small amount that remain that can turn out, in many cases, to cause relapse of the patient, and then the tumor grows out. And so we're very interested in being able to extract these MRD cells from a patient and be able to run drug tests on them. But it actually turns out it's not an easy problem to get these MRD cells. The markers for these cells are not perfect, both in terms of specificity and selectivity. And we are very interested in asking the question if the biophysical properties can be used as a way to enrich for these cells. And as kind of evidence for that, I'm going to show you what a pathologist would see. So what pathologists see, and here you're looking at these images here, these are lymphoma cells, and they have abnormal shapes. So as microscopy is currently used in the clinic, you can see these sorts of shapes, and that's what tells an oncologist how to describe what's happening. But what I wanted to try to tell you is that the microscope reveals, say, the size or the shape of the cells. But the size biophysically is related to its mass. And it turns out that its shape, in many ways, is related to the cortex stiffness of the cell. So what is the cortex of the cell? So the cortex of the cell is schematized here. It's the outermost layer of the cell. That includes actin filaments and myosin. And when the properties of that cortex change, it changes the shape of the cells. And in many types of cancer cells, that cortex is affected. And so we are looking at ways then to measure the stiffness in addition to mass and these other properties I showed you. And at the very beginning of the talk, I introduced this device for acoustic scattering, and that's what I'm showing you here. There's not enough time to explain fully how it works, but we published it. But the main thing is that this device vibrates in such a way to create very subtle fluid flows that actually cause the cell to deform. And when the cell deforms, the degree to which it deforms changes the effective stiffness of that whole cantilever, which then affects its vibrational frequency at a point that's independent from the mass. Again, we described this in the paper. I just wanted to kind of give you a sense of what we can do. And so we can now measure the mass and stiffness simultaneously from that same cell. And so this is an example that proves this. We're treating the cells with latrunculin, which disrupts the actin filaments in the cortex. And we can see the stiffness in our signal changes in response to that. And we can also see microscopically that the cortex is getting perturbed by this drug. And so a lot of the work that we're thinking about now in the lab is to validate these types of markers. And to do that, we really need to be able to take from the same cell the biophysical properties that we measure, but also have the transcriptome of that cell as well, to be able to really have full validation. And so this is work that's ongoing, but I can tell you that as I mentioned earlier, we have the way to collect these cells and then do single cell RNA-seq from them. And we've done some of this already based on cell lines, but we're now doing this on clinical samples that have a much higher degree of complexity than these simple proliferating cell lines. And we have developed the way now to be able to take these cells from the device and run them through a sorter and collect them, actually very much like a flow cytometer, and bin them, so that not only single cell measurements, but we can take bins of these cells and do bulk RNA-seq on different fractions, sorted biophysically. And so that's something also that we have recently been able to show. And I'll just give you an example. This is from a mantle cell lymphoma patient sample. These are normal lymphocytes, but if we look at the tumor cells superimposed, when we're plotting the stiffness versus the mass, we see some very interesting structure that is not the same as what we see from the normal cells. And so you can imagine if this green box here, we can take those cells and look at their transcriptomes and really see if we can validate those as particular types of MRD cells. We could have a way to go and enrich for them. Okay, so let me conclude with kind of a few key points here. What we really believe is combining these biophysical markers that I focused on here, but also with flow-based markers together, that you can have the potential to enrich for cells that would be really hard to do with either one alone. Another key point here is that ex vivo drug sensitivity testing assays, they have the ability to go beyond what genomics can do because they're integrative assays in terms of how the whole cell is responding to the drug. But they've been challenging for many reasons. And in the case of MRD, because these cells are very sparse and can rapidly deteriorate or reprogram when removed from the patient. But the ability to make these high precision measurements enables you to do acute testing on timescales that don't require proliferation and can minimize the amount of phenotypic drift that the cells could go through, maximizing the chance of the readout being able to be predictive of response of the patient. So that concludes my talk, and I will just acknowledge that I have been able to work with just an incredible team at the Koch Institute, as well as Dana-Farber in several different labs on this work, as well as funding sources from the NIH, as well as the Center for Precision Cancer Medicine and the Ludwig Center at MIT. So thank you very much, and I will stop there.